Hello, Namaskar, Khamba Ghani people. Welcome back to our channel, Professor Tiwari, and my name is Amrita. So today we are going to continue with the same topic of class 11, cell, the unit of life, and in that we are discussing the cell organelles. The next cell organelle that we are going to discuss today is the ribosomes. So let's continue with the ribosomes. Okay. First we look at the structure of ribosome. We will just draw a basic structure of ribosome. Okay, so this is a basic structure of ribosome. Now let's continue with this topic. So if I just talk about the ribosome, let's talk about the introduction of the ribosome first. Okay, so when we talk about the introduction of ribosomes, so ribosomes are actually, these are the smallest electron microscopic structure. I'm just writing the micro for this, microscopic structure. Okay. Now, next, see, these ribosomes are the smallest one that you will find in the cell organelles, or as a cell organelle, or in the cell you will find them as the smallest structures. These are actually the granulated. These are granulated structures. If you just see them, okay, they are actually being made up of two things, that is the ribonucleic acid, that is your RNA, plus proteins. Okay, so a ribosome is actually being made up of RNA and proteins. There are different type of RNAs are being there which contribute into them, and proteins, they collaborate together, they form the ribosomes. Okay, you will find the ribosomes in eukaryotes and in prokaryotes. So they are the characteristics of both the organisms. Alright. Next, what is? They are actually non-membranous structure, which means they don't have the membrane. As we have studied, the certain membranous structures were being there, like the your ER was being there, your lysosomes was being there, but these are not the membranous structures, these are the non-membranous structures. Okay, that is why they are common in the eukaryotes as well in the prokaryotes. Alright. Now moving ahead into this one, we will discuss about the history, a brief history of ribosomes, like who introduced ribosomes, who discovered ribosomes and when it was being discovered. Okay, so when we look at the history of ribosomes, actually the ribosomes were discovered in the year 1944 by a scientist named Cloud. Okay, so just look at this history of ribosomes. If you look at the history of ribosomes, so they are being discovered as a basophil bodies. They are discovered as basophil bodies in animal cell by Claude in the year 1944. Okay, and he named it as, and he called it as microsomes, because they were microscopic structures, so he called it as microsomes. Later in the year 1955 or 53, somewhere, some places it has been written as 53, somewhere it has been written as 55, there was another scientist called as George Palade. He discovered, with the help of electron microscope, he discovered these ribosomes and called it as ribosomes. So he was the first one who actually observed these ribosomes with the help of electron microscope and gave the exact structure, or called them ribosomes, gave the term ribosomes for it. Okay. Later ahead in the year 1959, two scientists, Tasso and Sau, they actually discovered the ribosomes into the nucleosomes. Okay, so ribosomes are also being found in the nucleosomes, and it was discovered by Tasso and Sau in the year 1959. Moving ahead more further, you might have heard the term polyribosomes or ergosomes. We will discuss these two terms later on in the section only. So in the year 1874 approximately, another scientist was two scientists were be there. Rich and Warner. They discovered polysomes or ergosomes. Okay, they discovered polysomes or ergosomes. They termed as polysomes is nothing but the arrangement of many ribosomes together on a mRNA. Okay, now so this was a history, like the first person that discovered the ribosome was the Claude in the year 1944 as a basal bodies in animal cells, and he called it as microsomes, which means the microscopic structures are being there. In the year 1955 or 53, somewhere it has been written 53, George Pallet discovered the ribosomes with the help of electron microscope. Okay, after that in the year 1959, sorry, this is not 1874, it is actually 1974. In the year 1959, Tassio and Sau discovered the ribosomes, presence of ribosomes in the nucleosomes. Okay, and in the year 1974, Rich and Warner discovered the polysomes or ergosomes. Polysomes means when many ribosomes they collect together and on the mRNA, okay, which means more than 10 or 20 ribosomes. All right. Now, moving ahead over here, we'll discuss the occurrence of these ribosomes. Okay, now what the occurrence is? See, as we know that we have already studied the basics of ribosomes in class 9th. So, ribosomes are actually the site of protein synthesis. Okay, so now next is occurrence. Okay, so basically ribosomes are the site of protein synthesis, and we know that ribosomes are being found in both in prokaryotes and in eukaryotes, both. But if we just talk about prokaryotes, in prokaryotes you will find them as in free form. Free form means you will find them as floating into the cytoplasm here and there, which means they are not attached to anyone. In the prokaryotes, they are only being found in the free form, whereas in the case of eukaryotes, if we just talk about the eukaryotes, then in eukaryotes you can find in the form of free form and bounded form. Okay, so you will find the ribosomes in the eukaryotes in free form and in bounded form. In bounded form, you will find on RER, that is the rough endoplasmic reticulum. You will find on them, and on the outer nuclear membrane. Okay, you will find it on two of the organelles, that is on the endoplasmic reticulum, giving it a shape of RER, that is the rough endoplasmic reticulum, and onto the outer nuclear membrane. Alright, now other than that, it is also important to study over here is that this whole ribosome is actually being made up of two subunits. Okay, so the ribosomes are actually being made up of two subunits, one and this one and this one. We will discuss like how they are being made up of, okay, later in its structure. So when we talk about the structure of ribosomes, so ribosomes are actually being made up of structure. See, as we have discussed this thing that ribosomes are being found in prokaryotes and eukaryotes both, so definitely their structures are a bit different over here. Now how they are being different over here? First we will say that both prokaryotes and eukaryotes, they have ribosomes, okay, but when we talk about the prokaryotes, I am writing about the prokaryotes first. So when we talk about prokaryotes have a ribosome of size of 70S. This S means Swedberg's unit, which means at what density, what is the density or size of the ribosome when you run the centrifuge, at what density it settles down, and what is the size of the ribosome. So Swedberg's unit determines that. Now if we just talk about it is 70S, so as I told you that the ribosome actually has two subunits, one is this one and one is this one. Okay, so whether it's a prokaryote one, whether it's a eukaryote one, both the organelle, or you can say both the organisms, the prokaryote ones or the eukaryote ones, have two subunits of ribosomes, okay, which is common in between them. When we talk about the 70S ribosome, then the 70S ribosome, I just divide it into two parts, that is your 50S and 30S. This one is your large subunit and this one is your small subunit, okay. So prokaryotes have 70S ribosome, that is the 50S and the 30S, alright. On the other hand, when we talk about the eukaryotes, then eukaryotes actually have 80S ribosome, okay, as the same Swedberg's unit. 80S ribosome actually has, it also has two subunits, one is 60S, that is the large one, okay, and 40S, that is the small subunit, okay. Now moving ahead into this one, as we have seen that the prokaryotes usually have the the ribosomes have two subunits, as the large one and the small one. What is the role of these large part of the, or the large subunit of the ribosome, or what is the role of the small subunit of the ribosome? Then actually the large subunit, when we talk about the large subunit, actually it attaches on mRNA, that is the messenger RNA that is coming from the nucleus in the with the transcription for the translation process. It attaches on the mRNA. Other than that, for the protein synthesis, other than that, large subunit also has enzymes. It also has enzymes which are used in during the protein synthesis. It contains enzymes which are important. We'll discuss what enzymes, what are those enzymes are, okay. Now talking about the small subunit, then small subunit actually attaches with, the large subunit attaches with tRNA, that is the transfer RNA, which will carry the amino acids, the codes that we call it, okay. The small subunit, it attach on mRNA, messenger RNA, okay. It is a site for messenger RNA, alright, fine. Other than that, the large subunit also, it is also responsible for the, it also has three sites over here. If I just noted down the three sides of this one is over here like this. I just draw it with three different pens so that you understand. Second is this one, and third is this one, okay. If you just call it, this is A site, this is P site, and this is E site, okay. So this is the three sites are being there, and they are being used as the site for the tRNA binding. Over here, this part is your mRNA binding site, okay. Now because we know that the large subunit has to bind to the, it has to bind with the mRNA also, or with the ER also, so it attaches with the ER with the help of a glycoprotein. Glycoprotein attaches the ER, attaches the ribosomes on the ER, and that will be called as ribofourin, okay. Whereas the small subunit uses the hydrophobic interactions, okay. It shows the hydrophobic interactions to attach to the ER, okay, alright. So now we will see that these subunits, the large subunit and the small subunit, they are actually been floating freely into the cytoplasm, or you will find them attached to the rough endoplasmic reticulum. Okay. Now moving ahead into this one, I will see more things over here, like types of ribosomes. So there are different type of ribosomes are being present. If we just talk about, we have seen the 70S and the 80S ribosomes are being there, but there are more type of ribosomes are being there. So let's discuss the types first. Types of ribosomes. Okay. Now when I say types of ribosomes, then we have 70S ribosome, the first ribosome that we're going to discuss. So you will find the 70S ribosome in prokaryotes. Okay, so it is, you will mainly find it in prokaryotes. Other than that, you will also find the same 70S ribosome in mitochondria. Mitochondria of eukaryotes. Okay, in mitochondria and called as ribosomes. Okay, and in plastids, and it is called as plastidoribosomes. Okay, all right. Now the next one is ATS ribosome. So we will find the ATS ribosome only in eukaryotes. You will find only in the eukaryotes cytoplasm. Okay. Next is 77S, 60S and 55S ribosomes. Okay. So there was a scientist called Levin Goodenough. This Levin Goodenough actually discovered this 77S ribosome, this 77S ribosome in mitochondria of fungi. Okay, whereas the 60S ribosome is actually being found in the animal cells. Okay, whereas the 55S ribosome you will find it in mammalian cells, mammalian mitochondria. Mammalian mitochondria, and this one is in animal mitochondria. All right. So we have seen there are three types of ribosomes: 70S, 80S, 77S, 60S and 50S. 77S is being found in prokaryotes, in the mitochondria of all the organisms, okay, like which is being called as mitoribosomes, and in the plastids of the plants, which is being called as plastidoribosomes. ATS ribosome is the only characteristic feature of the eukaryotic cells, which means it is only being found in the eukaryotes only, but into the cytoplasm. 77S, 60S and 55S ribosomes are being found in different organisms. Levin Goodenough, one scientist was being there, he found the 77S ribosomes in the fungal mitochondria, whereas the 60S mitochondria you can find it in the the 60S ribosomes you can find it easily in the animal cells of mitochondria, whereas the 55S ribosomes you can find in the mammalian mitochondria of the organisms. One more thing that is being there is being called as the biogenesis of the ribosomes. We will just quickly go through that biogenesis one. Okay. See, as we have discussed this thing that there is not only one ribosome, see what happens is there is a ribosomes which is being 18S, 5.8S and 23S. These ribosomes you will find in nucleosomes. Nucleosomes synthesize 18S ribosome, 5.8S ribosome and 23S ribosomes, whereas the 5S ribosome is synthesized in outside nucleus. It is synthesized outside the nucleus. Okay. Other than that, you have the polyribosomes. So polyribosomes, or you can say the polysomes. So this polyribosomes or polysomes, which means that you generally have more than 68 ribosomes you need to have. When more than 68 ribosomes are attached on an mRNA during the protein synthesis, then you will call it as a polyribosomes and polysomes. But it is not like that each and every ribosome will attach like a chain. Okay, they will actually attach at a distance of 90 nucleotides, which means might be 2 ribosomes are attached, might be 10 ribosomes or 50 ribosomes or 100 ribosomes are attached on one mRNA, but the distance between the 2 ribosomes has to be at least 90 nucleotides away from each other. Okay, now the last thing that comes up is the functions of ribosomes. As we all know that the ribosomes are the site of protein synthesis. Okay, other than that, there is one more thing has been there, that it also contains an enzyme called a peptidyl glycase, okay, which actually helps in the synthesis of, if you just look at the functions quickly, the last thing in the topic. So what do the ribosomes do? Ribosomes are site of, writing the short form, protein synthesis. Okay, so this is a site of protein synthesis. It contains an enzyme called peptidyl lyase. It contains again peptidyl lyase, which actually helps in the protein synthesis only. Other than that, ribosomes also help the newly formed polypeptide chain. This polypeptide, the polypeptide chain which has been newly formed, is actually protected by cytoplasm enzymes in large subunit. Okay, how does it protect? It actually protects it from degradation and transport it to ER lumen. Okay, all right, fine. So this is all for the ribosomes that we have discussed today. In the next class, we will discuss about the rest of the cell organelles that are being left. I think 2 or 3 cell organelles are being left. If you have any doubt in this topic, you can just drop them in the comment box below. Please like, share and subscribe our channel, and don't forget to hit the bell icon for more such updates. Thank you. Bye.